We're going to be continuing the history of genetics from the previous videos. This video will cover where we are now in the field and the issues that face us. One of the primary ways that genetics is being used today is in the use of GMOs, or genetically modified organisms. While GMOs can be described as artificially selected traits or species, they can be dated back to as early as 7800 BC. However, modern GMOs didn't start until 1974 with the first genetically engineered animal. Modern genetically modified seeds have been used for the last 20 years, but selective breeding has been used for hundreds of years. From there, it moved to hormones, plants, and food. These GMOs have allowed humans to produce more natural materials faster and more efficiently. This is helpful because by 2050, the world will need to produce 70% more food than it currently does to accommodate for an increasing population. As stated in the previous section, one of the main uses of GMOs is in crop manufacturing. Increased planting of genetically engineered crops has led to an increased use of herbicides. The definition of an herbicide is a product that kills certain targets while leaving the desired crop unaffected. They work by targeting key enzymes in the plant's metabolic pathway. This slows the plant's food production and eventually kills it. How are some plants then tolerant? Some acquire the trait through selection or mutation, where the plant is modified through genetic engineering. They have been blamed for some health effects, including skin rashes and death. Most plants are genetically modified to be herbicide resistant. The overuse of these crops has led to herbicide-resistant weeds and the destruction of surrounding areas. Therefore, it is important that they are regulated closely in order to protect our health and the environment. The use of genetics in this area is critical because without it, unchecked weeds would outcompete other plants for nutrients, space, and water that would inhibit useful plant growth. Another aspect of genetics that has grown in popularity is home genetics tests. They quickly became one of the most ubiquitous and well-known uses of genetics. There are many companies that provide this service, including 23andMe, Ancestry.com, and Invate. All of them offer similar services that range from the interesting to the medically significant. These tests can indicate if a person is at a higher risk of developing certain disorders, having certain physical traits, or even if they will enjoy the taste of certain foods. These tests are done by the client putting some saliva in a tube and then shipping it to their chosen company. The company then runs a polymerase chain reaction, PCR, which amplifies the DNA and allows for further analysis and interpretation. This allows the company to sequence the DNA and look for certain markers that would indicate specific risk factors. These tests have started to become bigger, and with them, some new ethical implications related to privacy have come up. One new use that has caused some concerns is that of the Golden State Killer. This has been a cold case for many years, but when his aunt did one of these tests and posted the results, law enforcement officials were able to identify and catch the original perpetrator. Although the apprehension of a serial killer is undoubtedly a good thing, this brought up concerns about how the data could be used, even for people who choose not to participate in such a test. As these tests become more powerful and universal, these issues will have to be addressed and regulations will have to be put into place. CRISPR is a genome editing tool that is precise in making genetic manipulations, including to the germline. It is the capability to remove, add, or alter sections of a DNA sequence. A genetics researcher in China has claimed to modify the first human embryos. He and his team accomplished this by using CRISPR to modify 16 embryos and implanted 11 of them to try to make a viable pregnancy. Their goal was to create identical twins that were immune to the HIV virus and therefore AIDS. They edited one gene in particular, known as the CCR5, which is known to cause the HIV infection. Although the test seemed to be a success, it has brought up many ethical issues. One of the public's main worries is that the editing of this gene will cause future problems that will be passed down the germline into future generations. Genetics is a constantly evolving field, and new innovations are constantly becoming a reality. As these new realities emerge, they bring with them many new challenges and ethical issues, some of which we will address in our next video on the future of genetics.